Very good day I bid to everyone who are watching this video. I am Jeffson Rusipin and I will be explaining about the chromatin, chromatid and chromosome. So the terms chromatin can either refer to two things, which is the first one is the structure of the chromosome, where the combination of the DNA and the protein that give the structure, or it can refer to the whole mass of multiple chromosomes in the nucleus, where the chromosome all jumble up together. Next will be chromosome. So basically chromosome always been referred to the well-defined one chain of DNA in this structure, and it only takes this type of shape at certain stage of its life when the cell is dividing. So the next term is the chromatid. So as you can see from the picture from the left, we can see there's a chromosome. So when the replication occurs, it will create itself another version of itself that is identical to the DNA, and it will look like this. But as we can see, they are identical to each other and attached to each other. But as I circle this one, this is a strand that we call as a chromatid, and there are also this one strand that we call chromatid. In other words, this is chromosome that have two chromatid. For more elaboration, after the mitosis cycle, they will be separate, and what we originally call it as one chromosome with two chromatid, you will now call it as two separate chromosomes.
Hello everyone, my name is Bavanraj and today I'll be explaining you all the term sister chromatids, homologous chromosome and tetrad chromosome. So stay tuned. Okay, so where do we usually hear this word or this term sister chromatids? Yes, exactly, during cell division. So before a cell can divide itself, it must first perform DNA replication, so that the new cells, each of the new cells receives a complete copy of the exactly same DNA. So I'll be demonstrating this by showing a straw as a chromosome, and this one at the center here is the centromere. So to receive a complete copy of the DNA, the two identical chromosomes must bind to each other, which means one of these and another one of these bind to each other, so that DNA replication can be performed, and it will result into this. This is a homologous chromosome. It is bound together by a centromere, and a full set of sister chromatids, which are all these, are created during the S phase of the replication process. We have three phases: G1, S and G2, and this is during the S phase. Okay, so now I'll be explaining about homologous chromosome. What is a homologous chromosome? Homologous chromosome is actually a pair of chromosomes we have in all our cells. So they are a set of our maternal and also paternal chromosome, which comes in and bind together. So for example, this is the chromosome from our mom and this is the chromosome from our dad. So in order for them to replicate, they have to be separated or splitted from their origins. So after splitting, when the process of replicating, when they combine or they bind into each other, is when they form a homologous chromosome, which is an egg-shaped homologous chromosome with a centromere. So now I'll be explaining about tetrad chromosome. What actually is a tetrad chromosome? Tetrad chromosome is two homologous chromosomes which will come together and crossover. This is called synapsis, and the crossover chromosome is called as tetrad. So the one as circled below, the chromosome moves closer together and synapsis occurs, where each of the chromosome binds together, and the brace shows that the tetrad chromosome, because tetrad means 4 and there's 4 pair of sister chromatids, so that's.
Hi and good day everyone, I'm Muhammad Fahim and I will be explaining about centromere, kinetochore microtubule and spindle microtubule. So these three plays an important role during cell division processes. First thing first, we have to know that this is a sister chromatid. The structure that is right in center of sister chromatid is known as centromere. So centromere holds together two chromatids. Centromere is a point on chromosome where mitotic spindle fiber will attach to pull sister chromatid apart during cell division. For example, during anaphase, you can see that centromere are now have moved toward centrosome, hence two sister chromatids are now separated. So now we are going to look at kinetochore microtubule, or also known as KMT. The main function of KMT is to control the movement of chromosome during mitosis and meiosis. As you can see right here, we have kinetochore protein on each side of the chromosomes. KMT will attach to kinetochore of chromosome. As you can see, each KMT will attach to each side of the kinetochore protein to ensure the chromosome to be aligned in equidistance. So finally, we are going to learn about spindle microtubule. Spindle microtubule will appear during prophase, where it pushes the centrosome apart. We have to also know that centrosome is known as the microtubule organizing center, where all microtubules are originated from centrosome. So remember, spindle microtubule will make two centrosome apart during the process of prophase.
Okay, Assalamualaikum. Alright, so in this video, we are going to over the basic difference between haploid and diploid and also somatic and gamete cells. So let's look at what are called somatic cells. And somatic cells are defined as a body cell of your body, non-cell cell. So example of your body: skin cell, brain cells, nerve cell, muscle, white blood cell, red blood cell, and this is what we mean by somatic cell. Now, let's see what inside the nucleus and looks like, whether it's a nerve, a skin, or a brain cells. Okay, the nucleus that we find chromosome work with this a stem rainbow structure that's called chromosomes. Now, arrange chromosome on a organized picture, right? So we're getting into a haploid cells. So notice, arrange, there's two chromosomes, okay? And number 1, number 3, number 10, number 18 have two chromosomes. So diploid cells are mean by cell pair, okay? So the chromosome are pair. So somatics, body cell are also diploids. Diploids mean they are pair chromosome, okay? In human, there are total of 23 pair of chromosome. So for a total of 46 chromosomes. So diploid cells are also created by the cellular division process known as mitosis. Alright, before we go a haploid cell, we need to examine what we call gamete. Gamete by definition what we call sex cells. Male and female have a sex cell. So gametes, male are produce sperm cell and female produce egg cells. Alright, so let's zoom into a nucleus of egg cell. So we have a bunch of chromosomes. Right, so the difference when the chromosomes are arranged from the biggest to smallest. Right, look at the picture. The chromosome 1 is the biggest, number 2 is a little smallest. Right, I hope you see the difference. Now we are coming what haploid cells. So haploid cells are a single chromosome. Okay, only one chromosome. Right, so you see number 1 have only one chromosome, number 15, number 17 have only one chromosome. So the chromosomes are not paired. Okay, a gamete cell are created by meiosis. Okay, we look at human. Human have haploid chromosomes, number of 23. That means it's a female cell. There are only 23 chromosomes. So remember, different of what we said a moment ago. If a cell are diploid in human, the diploids going to have total of 46 chromosomes, but the cell that are haploid only have half amount of 23 chromosomes.
Hello everyone, my name is Gayathri Ravi Chandran. Today I'm going to explain about cytokinesis, karyokinesis, chiasmata, and also crossing over. Let us go through one by one. The first term is cytokinesis. So what is cytokinesis? Cytokinesis is the division of cytoplasm of the parent cells into two daughter cells. Cytokinesis is the continuation of telophase, where a contractile ring will form at the center of the cell. Eventually, cleavage furrow will form and squeeze the cell and also split them into two daughter cells. Now let us go through the term karyokinesis. So what is karyokinesis? Karyokinesis is also known as mitosis. It is the division of the nucleus that occur in the mitotic phase. During the mitotic phase, there are multiple processes that take place which the duplicated chromosomes are aligned together, separated, and also move into two identical daughter cells. Karyokinesis divided into 4 stages. The first is the prophase, continue with metaphase, anaphase, and lastly is the telophase that result in the division of the cell nucleus. The next term is chiasmata. During prophase 1 in meiosis, homologous chromosome will be connected through synapses. During the synapses, the non-sister chromatids are connected at this point of exchange, a region called chiasmata, where it will hold the homologous chromosome together as a bivalent until anaphase 1. The last term is crossing over. Crossing over is occur in prophase 1 in meiosis when two non-sister chromatids of homologous chromosome exchange the DNA segment. The two daughter cells produced will have different recombinant DNA and will go through meiosis 2. In the end, produces 4 gametes, which is totally different from parent cell. That's all from me. Thank you guys.